We are recording again. Now, this has us starting off on chapter 15 from the very beginning. I'm going to skip around some because it goes through a bunch of the history that, again, you don't need to know, but a picture of a ribosome. So, translation. Let's go back to our analogy with grandma's cookie recipe here. And when you wrote down a copy of grandma's recipe out of the family book, you were transcribing it. Now let's assume that grandma wrote that recipe in a different language. And so now we need to translate that recipe into English. So you have to transcribe it. It needs to be written by somebody first, then you can translate it to a different language. So we went nucleic acids to nucleic acids. For that is DNA to RNA. Now we're going RNA to amino acids. It's a totally different language. That's why it's called translation. So 15.1 is, again, going through a bunch of the history that you don't need to know too much of. We're just going to talk about what their conclusions were and what things they kind of got wrong. So they found a bunch of strains that could not make the exact, or they could not follow the standard pathway for methionine biosynthesis. But if you gave them whatever step was broken, then they could go on from there. So they were able to figure out what the whole pathway was and figured out that each step involved an enzyme. So they had the idea that a single gene controlled the synthesis of a single enzyme. One gene, one enzyme hypothesis. This has since been modified to say that, well, enzymes are only one category of protein. Some genes encode other types of proteins that aren't enzymes. Then you have that some proteins are composed of more than one polypeptide. And that means that it's going to take more than one gene to be able to get a functional protein. So polypeptide, poly is many, and peptides have to do with the type of bonds that are formed between the amino acids. They're peptide bonds. And so you have a bunch of these stuck together. It doesn't mean it's anything functional. Once it gets into its 3D shape and has had all of its modifications done to it and everything, then it can be called protein because that's going to denote function. Number 3 is that you can have alternative splicing to allow one gene to encode many more polypeptides. And we've talked about this before with grandma's cookie recipe, that you might swap out the walnuts for raisins or vice versa, something like that. And then there is the many genes that encode functional RNAs. So it doesn't even necessarily get turned into protein to be useful. So slight caveats to the hypothesis there. But now looking at it, we use a genetic code that is going to be turning mRNA and saying which amino acid is going to be assigned to this spot. So these are known as codons, and it's recognized by the anticodon in the tRNA. So we've seen this already, the genetic code. This is a standardized one. And there's a couple that have some weird colors on them, and we'll come back and talk about them. But just know it is there. And now to show the whole picture, we have our DNA, which is up on top there, our template strand and our coding strand. And we're going to be always making our new strands in a 5' to 3' direction. So our mRNA that happens after transcription is also 5' to 3'. You have a 5' untranslated region. Then you're going to find your start codon, AUG. You're going to have all the different codons, then at the stop codon, and you end up with the 3' untranslated region. All of those different amino acids that got stuck together are then going to be the polypeptide that will become the functional protein. So they put the emphasis here that the start codon sets the reading frame for all of the remaining codons. So from that first AUG start site, you do not skip any letters as you're going through, and you do not double up. It's just read 3 at a time. Bam, 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 until you hit the stop codon. Nothing gets transcribed before the AUG, and nothing gets translated. Nothing gets translated after the stop codon. So don't tell me stop codon and then tell me what CCG stands for, because that means you don't understand what a stop codon means. So special ones. AUG is methionine. That's our start codon. AUG will also code for methionines within the protein. It's not like you restart each time. It's just after the first one, you just insert methionine and you just keep going. UAA, UAG, and UGA are all stop codons. They are going to end the translation process. And the code is degenerate in terms that you can have more than one codon that's going to code for the same amino acid. It's like proline, that is here second column, second row. CCU, CCC, CCA, and CCG all code for proline. And it's usually that third base pair in the set that's called the wobble base. It doesn't have to line up perfectly, and it still knows it's going to be a proline. It doesn't matter what the last one is. This codon chart is nearly universal. Like we might, for what we're doing, you can consider it universal. It does happen though that in mitochondria, instead of it just being AUG to get methionine, there's also AUA, and UGA, it's normally a stop codon, becomes a tryptophan. There are 2 other ones that are extremely rare that are usually found in like archaea, which are in extreme climates. You don't have to worry about it. Just know these 2 are not normally found in regular organisms that you're going to be working with in your lab. The fact that mitochondria have a different genetic code is another reason we think that there was some endosymbiosis, where a large cell essentially devoured a small bacteria that then became stable and hung out, because mitochondria contain circular chromosomal DNA, where the rest of our chromosomal DNA found in the nucleus is linear. Everything matches up closer to it having been a bacteria in the beginning. So just interesting things, but you don't need to know exactly what is coding for what. Like that's not going to be testable material. So when we talk about DNA being 5' to 3', we also have directionality to our polypeptide strands. It's always going to be between the amino group and the carboxyl group of the next ones. So one end, the first one made, is your N terminus, and the other end is the C terminus. The little group picture. So you have H3N, or NH3, is your amino group, and then the carboxyl group is the 2 oxygens and the carbon here. So you can attach these 2 together, and a water molecule gets popped out. And that bond is going to be a solid covalent bond. So in this case, we have AUG, so methionine went in. AGC, serine went in, and see, they were attached together as the peptide bond. You go on the whole way through. So there are 4 levels of structure in a protein: primary, secondary, tertiary, and quaternary. So the primary structure is just listing out all of the amino acids in order. Then secondary structures happen when the hydrogen bonds form between some of the side chains. You can get it spiraled up, called an alpha helix, or you get them laying side by side in parallel rows called beta sheets. So those hydrogen bonds that form the secondary structure, remember that hydrogen bonds are not as strong as a covalent bond. So back to where we talked about DNA being a double helix, the hydrogen bonds are what put the two strands together. You can unzip it and rezip it, no problem. Same idea here. Small temporary hydrogen bonds. Tertiary structure, though, is where you start getting covalent bonds. I mentioned we haven't talked about disulfide bridges and stuff, but know that it is the more stable, more functional form of the protein. So you don't hit the 3D shape until the third level of organization. Then sometimes you end up having to put two or more of those subunits together before you actually get a fully functional protein. So in this case, it's showing two of them attached, but they can be many more. I remember that histones are octamers that had 8 different subunits together. So in order to get into that functional 3D shape, they sometimes have to have extra proteins called chaperones that are kind of like an empty barrel that you load this string of amino acids into, and it's going to help fold it into its normal shape. You do not ever need to memorize all the amino acids, but I want you to understand how the categories work. Because when we talk about mutations later on, if you are swapping for an amino acid that's in the same kind of category, it might not even change anything on the protein. But if you swap to a amino acid that is from a completely different type, it's probably going to change that three-dimensional shape and therefore going to change the function of the protein. So the first ones that it talks about are littlest guys. They are non-polar. Remember, water is polar, and anything else is polar is going to like to interact with water. Non-polar ones don't want to interact with water, so they're going to be buried around to the inside of the protein. And the ones shown below it, there's 3 of them, have aromatic side chains that are quite large. So if you are swapping a glycine and an alanine, those 2 tiny guys, that might be totally fine. But if you suddenly swap a glycine for tryptophan, the biggest guy there, it's going to change that 3-dimensional shape that we mentioned before. The only ones I want to call attention to here are on the end. We already talked about methionine, and it contains a sulfur, and then there's cysteine that contains a sulfur also. It's particularly cysteine. Those two, the sulfur here, can interact with a sulfur on another strand to form disulfide bridges, and those are very stable covalent bonds. If you mutate out a cysteine and it was one that was used to form a disulfide bridge, you're going to destroy the shape of the protein. It's going to not stick together at all. Proline, right beside it there, is a weird one because if you see that it bends back and reattaches to itself, where everybody else just has that arch side chain going off, proline does an extra bend, and that makes it really good in like tight spaces. So where usually you have like a nice flowy ribbon, proline can have like a kink in the ribbon to help make a 90-degree turn if needed. The next classes are going to be our polar ones. So these again are ones that are going to like water, so they are want to interact with the water on the outside of the protein, hydrophilic. So the first ones there are neutral. They have no electrical charge. The ones in set D here are polar, but they're acidic. If you notice, they carry a negative charge, where the ones in set E have a positive charge on them. So if you swap a negative for a negative, that might be fine. Positive for positive might be fine. If you suddenly change out your negative charge for a positive charge, you're going to destroy everything. And then the non-standard amino acids, it just shows them, but you don't need to know anything about them. Just they're an option. All right, I already kind of talked about the secondary structures. And tertiary, quaternary, I already talked about. All right, you don't need to know anything about how they figured out which letters worked with which ones, but you can read about it if you want to. And the last, or not the last thing. So the structure and function of tRNAs, I'm going to come back to this when we do our RNA folding video, because it's easier when you're actually holding the shape in your hand as you do it. But realize that the tRNA holds an amino acid off to the side, and 3 of its nucleotides are going to be what interact with the codon. So our codon and the anticodon have to match properly for it to fit into the ribosome, and then that's going to allow the amino acid to join the polypeptide chain. There are some extra nucleotides. So it mentions more than 80 of these. So it has A, U, C, and G, but it's got some other weird ones here. You don't, again, have to know about them. We're just going to talk about them because, like here, an anticodon of I for inosine means that it can bind, it's going to show here in a little bit. Inosine can bind to all 4 letters equally. So that's why you can get that wobble base pairing happening, because the last one's an I, doesn't matter. Just know that there are ways to recharge the amino acids onto the tRNAs, because if you were to use them and they were one and done, it'd be very wasteful. You have synthases where you have the amino acid comes in, ATP for energy comes in, and then the bonds can be broken to release the energy to allow the tRNA to attach to the amino acid. And then the amino acid charged tRNA can float out. So here they talk about what letters can go with which ones. You don't need to worry too much about all of the ribosome structures and assemblies. Just know there's always a large subunit and always a small subunit. This is how it breaks down. But again, I'm never going to ask you all of these specific questions, so it's fine. So prokaryotes do this neat thing where you could have a single mRNA with many ribosomes working on it. So the mRNA here is in red, and technically this mRNA doesn't even have to be done yet. It could still be over here attached to the DNA being copied down. But a bunch of ribosomes can jump onto it, and they can each be making their own polypeptide as they go. So it's just a neat picture. You can't do this the same way in prokaryotes or in eukaryotes because of the nucleus. The ribosomes don't come inside the nucleus. They stay out in the cytoplasm. All right. And within our ribosomes, we have A, P, and E sites. A is the first one that is entered, beginning of the alphabet, beginning site, but I also remember it because it's the one that's holding us an extra amino acid. He hasn't been attached to the P site, which is holding the protein or holding the polypeptide. And then E is exit. That's once it's passed off as amino acid, it can then go off and get recharged again. You don't need to know too much about, I'm not going to ever ask you for all the different, like IEF2, anything like that. Just know that some things happen. Initiation has to have some helpers, elongation has to have some helpers, and termination has release factors that show up. There's also consensus sequences within the promoter region, or they don't have a promoter region, but these act like promoter regions. And where a promoter helped the RNA polymerase get set up to know where it was going to start laying down mRNA, this Schindel-Garno sequence is going to get the ribosome lined up so it knows where to start for AUG site here. Again, lots of videos for you to be able to just click through to practice with these. But know that there are initiation factors, there are elongation factors, there's release factors. There's special rules for prokaryotes and for eukaryotes to show you which AUG is. It's usually the first one, but it's not always. I'm not going to go through all these because, again, you can watch the video and they will do a much better job explaining it. Alrighty. So a little more information there. The last thing that the book mentions is about how antibiotics work. And a lot of the times they're coming in and they're doing things like blocking the translocation step. They're either stopping the amino acid tRNAs from binding to the ribosome. It can make it misread to produce abnormal proteins. There's lots of ways that we can change this pathway. And because the setup for prokaryotes is different than the setup for eukaryotes, that's how antibiotics can inhibit the bacteria without causing problems for humans, since we don't have a 23S rRNA. Okay. Any questions over chapter 15, at least on the sections that we discussed so far? Again, you're going to take an online quiz before Monday. You'll have to wait until I put it up. But again, it is assuming that you have already read the chapter, done the homework, that kind of stuff. It'll be a bit, it'll be like unit 1 that we had previously. So, any questions? If not, then I'm going to go ahead and sign off for this. Oops.